Thank you, Amy. I am happy to be here today to tell you all about some of the work I've been doing in the lab on gut bacteria and malnutrition. So first, to talk a little bit about how we define and assess malnutrition. One of the ways we do this is through the World Health Organization's definition of healthy growth, which they created using this multi-center growth reference study, which looked at thousands of individuals all over the world as they grew and developed after birth. And some of the things they measured in this study included the infant and children's weight and height in relationship to their age, and the ratio between their weight and their height. And from these metrics, they created z-scores that allowed these measurements to be standardized such that if there is a particularly low z-score for an infant or child, that indicates that there is malnutrition. And ultimately, the goal for all of this work is to help prevent or treat children that might be experiencing malnutrition such that they can shift back onto a healthy trajectory of normal growth. So one of the interests that my lab has is in the role of the gut microbiota, or the bacteria that live in our gut, in healthy human growth and development. And so we've found in my lab that there are bacteria that live in our gut as infants that shift over time, such that the bacterial composition of infant microbiota will be distinct from that of older children and adults. And part of this work was first pioneered by a former graduate student, Tanya Yatsenenko, and this is some data from Tanya, where she looked at individuals from Malawi, Venezuela, and the US over time of their life, which we have represented here on the x-axis. And then she looked at their composition of their microbiota. So on the y-axis here, we have a metric that indicates how dissimilar, which is more at the top, versus similar, lower at the bottom, a given individual's microbiota is to that to healthy adults. So Tanya was able to see that very early on in life, infants have a distinct composition in their gut of microbes compared to that of adults. But over time, they become more and more similar until they stabilize out in a composition about 2 or 3 years after birth. And so with this and subsequent work after that, we've been able to narrow down actually which microbes distinguish the infant gut microbiota from the child gut microbiota, and so on and so forth. And from these studies, we've been able to create our own standardized z-score, which we call the microbiota for age z-score, or MAZ. And we've been able to compare different MAZ scores for healthy versus unhealthy children. So we were able to have studies of microbiota from individuals in both Bangladesh and Malawi. And we looked at the MAZ scores for healthy children, which are represented here. And in these microbiota, the bacteria that we saw present were what we would like to say are age appropriate, so they're what we would expect given the age of the children. However, when we looked at infants that were the same chronological age but were experiencing unhealthy anthropometric metrics, we saw that they had significantly lower MAZ scores in both Bangladesh and Malawi. So even though these children were the exact same chronological age, the infants that were experiencing malnutrition had a microbiota that looked younger or immature than what we would have expected given their age. So this led us to some key questions. Given that we see this relationship between the composition of the microbiota and the health of the host, what extent does this idea of microbiota maturity actually impact host growth and development? Is there a causal relationship? And from that, would it be possible to alter the composition of the microbiota and subsequently alter the growth of the host? So to do this, we turn to our gnotobiotic mouse model. So I did this work in collaboration with Mark Charbonneau in my lab. And what Mark and I did was to select a number of different fecal microbiota from Malawian infants. Half of them were from healthy infants, and about the other half were from unhealthy infants. And we took each fecal sample and transplanted it into germ-free mice. And then we followed the mice after they were colonized to see how they would grow. In addition to watching them for about 5 weeks after colonization, the mice were fed a diet that we like to call M8, or Malawi 8, which is a prototypical Malawian infant diet representing the complementary foods that these infants would be eating. So as I mentioned, we assessed the growth of these mice And we did that through body weight and measuring their body composition through a quantitative MRI, which allowed us to measure their fat mass and their lean mass. So what did we see? We saw that mice that had been transplanted with a healthy infant donor microbiota grew more than those that had been given an unhealthy infant's microbiota. And this was both by total body weight and lean mass gain, suggesting that there is, in fact, a causal relationship between the composition of your microbiota and the growth of the host. So next, we wanted to find a way that we might be able to alter the microbiota composition and see what would happen to host growth. So the first approach we did for this was something that we like to call the battle of the gut microbiota, where we would have two groups of mice, one colonized with an unhealthy infant's microbiota, and the other colonized with a healthy infant's microbiota, and then co-house these two groups to allow the two different communities to be exposed to one another and change. And that from that changed composition, we wanted to see what might happen to host growth. So the setup of this battle was as such, where as before we had mice that were colonized with a healthy infant's microbiota, or mice that were colonized with an unhealthy infant's microbiota. And then after a period of time, we took these two groups of mice and co-housed them together to allow the two different communities to be exposed to one another. As controls, subsets of each group were held back such that there was a healthy control group that had only ever been exposed to that one community, and vice versa, mice for the unhealthy control group that had never been exposed to the healthy. And as before, these mice were fed this prototypical Malawian diet, M8. We colonized them at the same age as before at 5 weeks. 4 days post-colonization, we co-housed our experimental groups. And then we followed the growth of these mice for a total of 21 days post-colonization. So what happened with these mice was as follows. So this is looking at the lean mass gain of these mice at the time of sacrifice. And what we saw was that in both groups of co-housed animals, there was a significant increase in the gain of these mice compared to that of our unhealthy control. But there was no significant difference in the lean mass that they had gained compared to that of the healthy control. So something in the process of co-housing seemed to have enhanced the capacity of these mice to gain lean mass. One thing that we also observed was looking at the proxy of the diversity of the bacteria present within the gut. So this is the Shannon index, which is a proxy for the how many different types of bacteria are there. And we saw that there was no significant difference between our 4 different groups, indicating that it wasn't simply a matter of there being more bacteria present in our co-housed groups that may have caused this increased growth, but there might be particular bacteria responsible for this change. So going ahead and looking at the microbial dynamics that occurred within this co-housing experiment, I have a heat map here. So each row represents a given bacterial species, while each column represents a single mouse on a given day, and these are grouped by their experimental groups. So here we have my healthy control mice, and you can see throughout the totality of the experiment here, all of the bacteria that are highly abundant, and all of these bacteria that are highly abundant here are distinct from the bacteria that are present in my unhealthy controls here. So there's a whole swath of these bacteria that are present in the healthy that are completely absent in the unhealthy, and then vice versa. Looking at my healthy co-housed group, with the arrow indicating the time of co-housing, you can see throughout this experiment as well, even after co-housing, this community remains largely stable. When we look at my unhealthy co-housed group, we can see prior to co-housing, as we would expect, the bacteria present in the guts of these mice resembles just like that of our unhealthy control. However, once these mice were co-housed, I saw a large invasion of many of these bacteria from the healthy community into the unhealthy community, and with that, the displacement of many of the unhealthy microbes that had previously been very abundant. So this brought on another question of what was really leading to this increased lean mass gain that we'd seen within the mice. Was it the addition of all of these taxa from the healthy microbiota, or was it the loss of all of the unhealthy taxa that had been previously abundant? So to more directly test this, we proceeded to take the unhealthy microbiota that we'd been working with and then culture out some of the microbes that we had seen that had invaded the unhealthy community. And I did this in collaboration with Tarek Salih in my lab. And so having cultured out about 5 different of these healthy community-derived invaders, I simply added them to my unhealthy community and wanted to see what would happen. And I saw that the addition of these healthy-derived strains, both by total body weight and lean mass, caused an increase in growth compared to the unhealthy control, showing that the addition of these healthy taxa seems to be sufficient to increase host growth. Taking a closer look at what microbes actually colonized in this experiment, though, interestingly, I only saw 2 of the 5 bacteria, Arumnococcus nevus and Clostridium symbiosum, actually successfully colonized the unhealthy microbiota, which you can see here. So we have the Clostridium symbiosum here, which took up about 2 or 3% of the community, and the Arumnococcus nevus, which took up about 20% of the relative abundance of this community. Also noteworthy was that while these added 2 taxa did take up a portion of the community, none of the original species that had been present in the unhealthy community were completely displaced, suggesting that it really is the addition of these bacteria that's sufficient to cause this change in host growth and not necessarily the loss of the unhealthy derived bacteria that were necessary. So to sum up some of the points that I've shown you today, we have evidence that microbiota from unhealthy infants confer less growth to the host compared to that of microbiota from healthy infants. I also showed you a co-housing experiment where when an unhealthy and healthy community were co-housed together, the healthy microbiota was able to win, quote unquote, over the unhealthy microbiota, and both groups of co-housed mice were able to grow more. Finally, I saw that addition of this R. navis and C. symbiosum from the healthy microbiota were sufficient to increase growth of the mice. And so with that, I'd like to thank everyone in my lab, particularly Mark Charbonneau and Tarek Salih, which helped a lot with this work, and I'd be happy to take any questions. Yeah, so there's a bit more known about the Arumnococcus nevus. So it seems to associate pretty closely with the mucus lining of the gut. So it can metabolize lots of carbohydrates and things like that, which might be one way in which there could be more energy that could be made available to the host. But there really hasn't been too much done with those 2 species, ultimately. Mm-hmm. Yeah. Mm-hmm. Yeah, that's a great question. So the M8 diet is not particularly healthy. So the way it was designed was to not only model the ingredients that are consumed in complementary foods in the Malawian infants, but also the nutritional content. So for both the infants that are often being fed in Malawi and the way the diet is modeled in our mice, there are many nutrient deficiencies across the board. Mm-hmm.
When you take in physiological food, you possibly add in the most best bacterial strain and engineer that in the house where you keep bacteria against one another and add the best one in. Could that be the most beneficial bacteria for mouse growth?
Yes, I think that wouldn't necessarily, simply because the mouse would, because the bacteria would be able to outcompete others in mice, that wouldn't necessarily mean that it would promote growth. There could be pathogens that could easily outcompete more healthy, quote unquote, bacteria. But what was really interesting here is that we were able to find the sort of the intersection of that, where we found microbes that were able to successfully colonize and promote growth. But I think it would really have to be on an individual basis, strain by strain, if they would have that capacity. Mm-hmm. So yeah, the metric that we have is just sort of for lean mass across the whole host. We didn't specifically break it down into more compartments beyond that, but a lot of that would presumably be muscle, but that's not necessarily 100% the case. There's other tissues that read as lean mass in the way that we were measuring the mice. Mm-hmm. Yeah, I haven't done that experiment. I would hypothesize that they would, because there is such a dramatic rearrangement of their composition. I would suspect that many of the unhealthy bacteria would not necessarily come back up. But yeah, we haven't done that experiment for sure. All right, thank you.